Okay, so your syllabus is your guide and it will be your reference point. Understand the concepts. You should understand the concepts, then you will memorize. Sometimes you cannot understand the concept if you are reading a book. So watch videos. Videos are very important for you to understand the concepts. Then you can memorize majority of the information. Not everything you should memorize. Memorize the basics, the most important things, the major things. Then you can solve past papers. Past papers are the most important. So you have to be systemic in studying for your exams. Leave the last 4 weeks for you to study the past papers. So 4 weeks before your exam, you have to put it to solve past papers. Print them if it's possible, because writing on the paper will make you memorize better, and it will give you the atmosphere that you are in the exam. Time manage your paper. Even if you are not in a test, time management is very important. It will enable you to solve your past paper faster. Put for yourself 45 minutes to solve the whole paper. And this is when you finish studying your subjects. Mark every paper following the mark scheme after solving it, and revise the subject if you lose mark. If you feel that you are losing a lot of marks, so this means that you have knowledge gaps, so you have to fill these gaps. Okay. There are very important points. Most of the students, they are doing mistakes not because the exam or the test is very hard. It is about not paying attention or not writing precisely. So you have to read the question carefully, pay attention to the keywords, to the drawings and charts. Depend on your knowledge that you got during studying the subjects. Apply different strategies like elimination when you have a multiple choice. So eliminate the most wrong answers and using the keywords. Uh, I did a research, so I found 10 most common IGCSE questions, and these questions, most of the students are being confused in solving these questions. Although these questions are not very simple, to be honest, but it's simple. You can get higher marks by knowing what to do and how to write the answers. One of the most common questions is movement of substances. The most popular question coming in every year in every past paper is to define diffusion, osmosis, and active transport. You should know the difference between these three. Who want to give me the answer of this? Okay. Dana, you want?
Okay, so it's the movement of particles. It's the movement of net particles from high concentration to low concentration.
Yeah. Thank you very much, Dana. So movement of particles from areas of higher concentration to areas of lower concentration. If you said movement of substance, it's wrong. So you should use the word particles. You have to say that it's from areas of higher concentration to areas of lower concentration. Or you can add against or down, not against, down the concentration gradient. Who knows what's the meaning of concentration gradient? Ahmed. Ahmed Abdullah. Do you know what's the meaning of the concentration gradient? Okay. What about you, Nana? Okay, who wants to answer? Okay. So I will tell you that diffusion is the movement of particles from areas of higher concentration to areas of lower concentration. You will get the 2 marks if you say this. You can add down the concentration gradient. If there is area of a higher concentration and another area of a low concentration, so you will have a concentration gradient. And you know that the rate of diffusion will increase of this if this gradient is higher. So if there is a very difference in the concentration, so the rate of diffusion will be very high. What about osmosis? You have to tell that osmosis is the movement of water molecules, not any particles, not any molecules, water molecules from a dilute solution to a more concentrated solution through a partially permeable membrane or a semi permeable membrane. You have to say or to write all these 3 points and you will get 3 points. Movement of water molecules from a dilute solution to a more concentrated solution through a partially permeable membrane. And this is the area most of the students are missing, they are making mistakes. Dilute solution, it means what? There is another word for it, more water potential, because we are concerning about movement of water molecules. We are not concerning about other particles. What concern me is the water molecules. So more water potential means more water in it. To allow water potential, and it is the same meaning. So dilute solution, it means more water potential. Concentrated solution, low water potential through a partially permeable membrane. And this is where you will have questions asking you about turgid pressure, about flaccid cells, and they will give you questions under the microscope what's going on. So you have to answer the isotonic, the hypertonic, and the hypotonic solutions. For the active transport, it's the movement of particles from areas of lower concentration to areas of higher concentration using energy from respiration. Because this is a process, it's against the natural things to happen. It's from lower concentration to areas of higher concentration. And this is the way that the living cell is producing or getting the minerals needed for a lot of biological reactions. This is one of the most common questions. The other one is arteries versus veins. This is from the circulatory system. The uh, most popular question is to compare between the arteries and the veins. You have to do what? You have to name the thing that you are comparing. So you cannot say they are thicker walls, they have thicker walls, they are more elastic. You have to choose one. So you can say, you should say, arteries have thicker walls, more elastic tissue, smaller lumen, no valves, while vein have. And you will get the whole mark. 3 points for this type of questions. So you should use a comparative language because you are comparing. You should do what? Name the thing that you are talking about. Don't say veins or arteries. Don't say they. You have to name it. Arteries are blah blah blah, or veins are blah 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 according to your knowledge. This is the second question. The third type of questions: explain how human lungs tissue allows efficient gas exchange. You have 4 points for this question. And most of the students, to be honest with you, are making mistakes. I don't understand why. It's very obvious, it's very clear. So you have to talk about what? Alveoli, because that gas exchange, where it's happening in the alveoli. You have to talk about the concentration gradients of gases. You have to talk about capillary walls and giving reasons, not only describing. You will not say alveoli are branched and they are very close to the arteries so they can get. No, talk about giving reasons. Give me the description and give me what will happen. How effective will the alveoli be in gas exchange? So you will say the alveoli provide a large surface area. Alveoli walls are 1 cell thick for what? Give me how effective is this for short diffusion pathway. Alveoli walls are moist for gas to dissolve. Capillary wall is 1 cell thick for a short diffusion pathway. The flowing blood maintain a concentration gradient, because we know that in the capillaries we have arterial blood and venal blood, and we know that the arterial blood, it's oxygenated and the venal blood is deoxygenated, so the concentration gradient is kept around in the surrounding around the alveoli. What are the mistakes that the students are doing in this? They will say that alveoli cells are 1 cell thick. It's not the alveoli cell. Alveoli walls, not the alveoli is 1 cell thick. It's the walls of it. So watch what you are writing. The other question, the fourth type of questions, it's talking about absorption in the small intestine. Explain how the structure of the small intestine is adapted for efficient exchange of substances. It's the same story of the alveoli, but it's a different one because it's in the intestine. But I'm talking about the surface area, how effective the adaptation in these cells are providing the best function. Villi provide a large surface area. Villi have a thin wall for short diffusion pathway. Villi have many capillaries to absorb glucose. The blood supply maintain a diffusion gradient also. The villi have lacteals to absorb fatty acid and glycerol. Lacteals are long to provide a large surface area. You have to mention all of these and you will get the 4 marks for this. The fifth type of questions is what? It's the genetically modified bacteria. Genetic modification. Genetic modified bacteria are very important in genetic engineering and in applied genetic engineering. This is very important for all the levels. A levels are concentrating a lot on these subjects. So you have to know the steps. You have to know that the first step, they will give you a question. Describe the stages that are used to genetically modify a bacterium that is able to manufacture insulin. It's insulin in this case. Maybe he will ask you to manufacture another hormone, another protein. So, talking about genetically modified bacteria, it's the same story with using a certain substance or hormone or protein to manufacture. So, first of all, you have to know that the first step is to isolate the bacterium plasmid. Plasmids are vectors, meaning that they are carrying the things. Open the plasmid using a special restriction enzyme. Extract the human gene. Here it's insulin, so you will use the same restriction gene that you used to do what? To, to open the plasmid. Why? You will open the plasmid. How you will open it? You will open it with a restriction enzyme. It's like scissors, but it's cutting the plasmid in certain areas. You will use the same enzyme to cut or to extract the human gene using this same restriction gene. Why is this very important? Because it will leave the plasmid with sticky ends and leave the DNA that you extracted, because the gene is part of the DNA. So you extracted the DNA that you want to stick it on the plasmid. How you will stick it? You should use a glue, and here the ligase is your glue. It's the enzyme that you use to join the plasmid with the gene that you extracted from the DNA. Then a recombinant plasmid is formed. This plasmid will be reinserted into the bacteria using a bacteriophage type of virus that it will cause to insert the plasmid again in the bacteria. Any question? Who wants to ask any question, please? Nana? I cannot hear you clearly, miss. Nana?
Hello, ma'am.
Yes, hello.
Can you change the slide? It is the displayed one, only the first slide.
Okay, now?
It's only the first slide that appeared.
Okay. So it's biology
Click
only?
on slide,
Now?
number 2. No, you did not change it. You can just click on the slide number 2
In the PowerPoint.
in the PowerPoint, please.
Yeah, I'm doing this.
But only the first
Slide appears.
slide appear only.
Okay, so I will, okay, let me stop sharing and I will share it again, okay?
Thank you, thank you.
Yeah, welcome. No, this is not the one, sorry. What about now?
Not yet.
Not yet. Okay. Has stopped. It stopped. Okay, no problem. Still, miss?
Yes, ma'am. Yes.
Yeah, it's working now?
Yes.
Thank you very much. Thank you. I'm sorry. I'm very, very sorry. So, do you want me to repeat genetically modified bacteria?
No, it's clear.
or it's clear?
It's clear.
Okay. So you have to mention what? The plasmid, the vector, open the plasmid. It's logical. So be logical. If you are being logical, everything will be solved and your self-confidence will be very high. If you know, I know this. Most of the students, they know this, but they cannot put it on the paper. This is the issue. So you have to go through these steps. You have to mention the plasmid. You have to mention that these plasmids are vectors. We are using them to manufacture the protein or the hormone that been asked about. Open the plasmid using restriction enzyme. Extract a human. We want to get the code for this gene, for this hormone. So you will take particular area of the DNA to component or to bind it to the plasmid, and we need something to put these pieces together. So ligase is used, then a recombinant plasmid is used, then we will reinsert it into the bacteria, and when the bacteria is reproducing, it will produce insulin with it. Okay, so this is the fifth most important question. The sixth one is talking about designing an investigation. Most of the students also, they don't know from where to start. There is one way to use it to remind yourself with all the things that you have to talk about. You will use something, we call it COMS. For an example, design an investigation to find out if the color of the light affects the amount of a fruit produced in the plant. I don't have any more data. They asked me to design an investigation to find out if the color of the light affects the amount of fruit produced in plant. So what I want to measure, what they ask me to find out, what are the things that I have to change? What are the things that I have to keep the same for my experiment or my investigation to be reliable? So we will use COMS. C means change, what is changing, what you are changing. O, organism, what is the organism that you are using? Is it a bacteria or plant or what? Reliability, how many you are repeating your experiment, or how many organisms you are using, for how long. M, it means measurement, what you are measuring. Here we are measuring the mass or the amount of the fruit produced in plant, so the mass. And what the things that you are keeping constant, you are not changing. So for this question, design an investigation to find out the color of the light affects the produced in plants. So if you please, I need to hear what you will say. Yeah? 16. Okay, so for this experiment, who wants to share me what you will write in your test? What you will write in your test? What about you, Ala? Ala?
Yes, miss.
Yes, miss. What you will write in this type of questions?
I will use the terms like change and the organism's reliability measurement
Mm-hmm.
and in steps, right? According to the question.
Okay, so tell me what you will do. Okay, I will take a number of plants and I will put them under
Repeat
what?
the organism. I will choose the organism and repeat the experiment.
Okay, repeat the experiment, but you have to be precise. You will not say,
Okay.
I will bring a plant. No, you have to be precise. I will grow tomato plants of the same age under what? Different colors of light. So, what I will change? I will change the light color. So, I will use red, blue, green, and yellow lights. And we have these. I will grow
Okay.
tomato plants of the same age. They should of the same age under the same light, the same age, of the same size, of the same species. So you choose the organism that they have the same age, the same size, the same species. So we are using tomato plants. You will do you will
Reliability.
do what? You will grow 5 plants. This is the repetition for reliability. We said that you have to repeat your experiment many times for your results to be reliable. Grow 5 plants under each color. Then what you will do? Measurement. Record mass of tomato. You cannot say I will record amount of tomato. You have to be particular. You have to be precise. Particularly, I'm using the mass. I'm weighing how many tomatoes or how heavy the tomatoes that are produced from these plants. I will do this for 1 month. This is with the measurements. You will record the results. What you will keep stable? You will keep the same light intensity. So you will put it
Light
on a
intensity.
distance that it's far from the plants for the all the colors. You will keep the light intensity the same. You will not use a bulb with higher light intensity. So you have to keep the light intensity the same because I'm not measuring the light intensity. I'm measuring what? I'm measuring the effects,
The mass of
not the effects of the color of the light, not the light intensity. I want to know. There is a very important thing also. We will irrigate the plants with the same volume of water. There is a very important thing in designing and investigation. You have to set a control group. Who can tell me what is a control group? What is a control group? Okay, I will give you the permission to talk. Who's this? Do what you love. Okay. Okay. What is a control group? Who wants to share? Okay, what about you, Nana? Sorry, I can't see all the participants. Okay. The control group is a group that you keep everything constant or not changing. But for here, you are using different lights, colors, different light colors. So what we will use? We will use the natural one. We will use the visible light here, and we keep changing the colors. So this is the control group that it's undergo the natural conditions, because we are studying the effect of different colors of light, and the thing that we know that all the plants are under the visible light, but we want to investigate, is there a effect an effect of the light itself? So in other words, is there an effect of the wavelength? And mostly the wavelengths are different, they are affecting actually. Okay, this is very important, and the most important thing that you have to state your variables. You have to know which variable is the dependent one, which variable is the independent, and which one is the control variable. For this investigation, you have to name the dependent and the controlled variables. There is a question. A student conducted an experiment to find out how the type of light bulb affects the mass of fruit produced by plants. Plants are grown under lamps for 3 types of bulbs are used: incandescent, fluorescent, and LED. This is another experiment. Make an investigation. But here they are asking you what are the variables in this experiment. You have to be precise, like we said. Okay, let me explain this in simple words. The dependent variable, the variable that you are measuring, that you what? That you want to measure, yeah? Independent variable, the thing that you are keep changing, and here in this experiment, it's what? It's the type of light bulb. What are the controlled variables that you are keeping the same? You are keeping the temperature, you are keeping the light intensity, you are doing what? You are using the same species of plants, the same age, the same size. These are the controlled variables. And most of the students, they are confused between the dependent and independent and the controlled variables. The controlled variables, what we will keep the same for all the experiments. The independent variable, the one that you are changing, it's not affecting, it's the factor that you want to change. While the dependent variable, we want to see, is this variable depend on the changing that we are doing? So it's the variable that we are measuring. The variables are very important, not only to solve this type of questions, because when they ask you to draw a graph, you have to know where is your dependent variable and where is independent variable. And this we will come within few minutes to solve this type of questions. Okay. When you solve variable questions or questions talking about variables, the independent and dependent and the control, you cannot say the amount, be specific. You will say the mass or the volume or the temperature. When you are talking about light here in this question, you have to mention light intensity, is it light type? And this is what we are talking about here. It is the light type that we are using it as the independent variable. Don't mention and say substance. Whatever you have, name it. If they are talking about glucose, say glucose. If they are talking about starch, say starch. Here we are talking about the mass of the what? The mass of the fruits. So the mass of the fruits, does it affected by the light type? And you will say that the mass of the, depending on your results, you will say that the mass is affected or disaffected by the light type, depending on the experiment that you have in the question. And the other thing that a lot of questions coming about is about the reliability of the results. A lot of questions is telling you, give two reasons why her results were reliable. Why the student doing is right. You have to think about the most important things. How the experiment could be reliable. If the experiment repeated for many times. If you are doing or setting the variables right and the results are recorded under same conditions and the steps that she carried out to keep the experiment very relevant. One of the questions here is talking about what? There is a question that a student want to compare the amount of starch in different food liquid samples. So what she did, the student, she brought 3 test tubes. She bought 3 centimeter cubes in the of each sample in different test tubes. She repeated the experiment for 3 times. She added 2 drops of iodine solution to each test tube. Then she compared the intensity of the color change on a spotting tile. She used a tile to compare the intensity of the color. You know that for finding starch or indicating that there is starch in a food sample, you will use the iodine. The color of iodine is brown. It will change from which color? It will change from brown to blue or dark blue if starch is there. So when she did this, what she did? She used 2 drops only of iodine solution. She repeated the experiment, so her experiment is reliable. She added 3 centimeter cubes of the samples. She used the same amount of food sample. So talking about what she did, don't say she did repeats. No, you have to mention what she did. She used 3 test tubes for each sample, giving repeats. She used the same volume of food sample for each test. These are the things that you have to mention. The other question you've been asked to do or you will be asked to do is to plot a line graph. Am I very fast or it's okay for all of you? This is a very important subject because most of the students they will be lost in doing this. Some of the questions will ask you to complete the data. Your data will not be ready most of the time. You have to complete the data. So we'll use something and this will help you to follow these points. We will use something, we call it SAPLOC. SAPLOC. So the first one is the scale. The A is for axis, P for points, and L for line, U for units, and K for key. Okay. When the question will ask you to draw a graph, Can you see my screen? Okay. First of all, the scale. You will use half of the graph paper that is provided for you. You should use half of the graph. Don't make your drawing very small or make it very big. So use the half of the graph paper. Look at the greatest numbers that you have because you want to put a scale. So for an example, if you have data ranging from 2 to 10, you will use the skip counting 2, 4, 6, 8, 10 till you reach 10. Sometimes you will have the first number maybe 2.5. So use 2.5, 5, 7.5, and etc. So your scale should be the same. The second thing that you have to care about is the axis. You know that in each graph we have the x-axis and the y-axis all the time. The independent variable will be on the x-axis and the dependent one on the y-axis. What does this mean? Independent one that you are changing. What you are changing? Like the previous question, what we changed? We changed the type of the light. What we want to measure? We want to measure the what? The mass of fruit produced if we are using different light types. I will give you an example. Wait, but I'm giving you the rules. So use half of the graph, label your axis, the independent on the x-axis, the one that you are changing, the thing that change, the thing that you want to study, and the not the one that you want to study, the thing that you are changing. On the y-axis, you will put the thing that or the variable that you want to measure. P, plot your points carefully with an X. Don't put a very big full stop or a very big X. Choose it and use the data carefully and plot your point carefully. For the line, which is the L, connect the first point with the coming point, with the second point, using a ruler. Don't connect it using your hand. Use a ruler, please. Most of the marks are lost because you are using your hand to connect the whole points from the beginning till the end. Each 2 points, use your ruler. And don't connect to 0 unless you've been told to. Don't connect your graph to the 0 or may extend it. Don't use it. Use the data that you have and connect the points and leave it. That's it. If you have units, include the units if you have with the variables. Sometimes like the type of light, you can add the types that you've been used. For the mass, is it in grams, in kilograms? So you have to do it. The last point is the K, which is put a key if you have more than one set of data. This is for drawing a graph. And it's the same if you are drawing charts. Okay. Dr. Aisha? Okay. Who has a question about these things? Okay, I cannot hear you, miss. I cannot hear. Who's that? No, he is talking.
Hello students, how are you? If you have any questions and the internet is not working properly, please type in the chat.
Thank you, Dr. Ash. Okay, so I will continue so we can solve questions. I prepared some of the questions that talking about the most important questions, tips. For the bar chart, you know the bar chart. It's like columns, and you have also the histograms, but mostly they will ask you to draw a bar chart or a line graph or a histogram. The histogram is talking about continuous data. The bar chart is talking about discontinuous data. Also, we will use the subblock. Sometimes you will leave the K, sometimes you will put the K. It depends on the question that you have. And as we said, is for the scale, axis, points, you have to draw the bars of exactly the same height. You will leave a space between each bar. So make your scale that you will leave uh, a space between each bar. You will use to draw you will use a ruler to draw the bar. Don't use your hand, please. Any unstraight line, you will lose mark. So your lines should be straightened. Put the units and put the key for your graph or chart. Label it. Okay. Also, you have to use the half of the graph paper, put even scale depending on the biggest number you have. Draw the axes, label them, write a key and a title of your graph. This is one of the questions. I think it's clear for all of you that a student want to investigate the factors that influences the activity of soil organisms in a woodland. She decided to study one species of woodlouse, which is a small animal found under stones and rotting wood, damp and dark places. This is the picture of this organism. She used a trap to collect organisms in the woodland during the daytime and during the night time. She counted the organism collected before releasing them. And these are the results. There is the day time sample and the night sample, nighttime sample. What she want to study? She want to study the what? The wood lice. She chose to make a sample during the daytime and another sample during the night time. The question is asking you from the daytime sample she produced a table results. You have the results for the day sample. Wood lice, it has 7 spiders, it has 1 centipedes, 2 beetles, 3. And here you notice that there is a tally mark using. So you should do the same for the night time sample. You will put the tally marks, then you will put the number. So let's do the question. Count them. Daytime, done. That's it. We will do the night time sample. Is it clear for you that you can see? Okay. These are the wood lice. Count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15 of them. How many spiders? 2. How many centipedes? 1. How many beetles? 2. And you will put it as tally marks and as numbers. So here you will put 1, 2, 3, 4, and 5. 1, 2, 3, 4, 5, and also it's not that much clear. My control is 1, 2, 3, 4, and 5. You know that for the tally marks, every 4 and you cross, this is the 5th one. So the number is 50. And you will continue with the other organisms. How many spiders? We said 2. How many centipedes? 1. And how many beetles? We said 2. Okay, this is a ready solution for it. Sorry, clear. So you have the solution 15, 2, 1, 2. The second step will do what? What we will do? We will graph a bar chart. They said a bar chart. Okay, how you can animate clear. Okay. So the second step, what we said. So the first step is what? You remember? And drawing the bar chart. The first step is what? You will keep the scale. The second one is to do the axis. You have to label the axis. Here the x-axis and here is the y-axis. So what you will label the x-axis? What you will put? Okay. Okay. Can you raise your voice, please? Who's that? Who's that? Ahmad? Is it Ahmad? Okay. So the first step, you will label your axis. You will put the independent one on the x-axis. So here what we will put? We will put the type of organisms on the x-axis. So here you will write what? The type of organisms. What we have wood lice, then try to arrange your data depending on the table that you have. Okay. Okay. So we will put wood lice, spiders, centipedes, beetles.
Excuse me, I would really appreciate if everybody mutes their mic because we can hear a lot of noise from somebody's mic. So please mute your mic, please. Thank you.
Thank you, Dr. Aisha. So the first step, you will put the type of organisms on your x-axis. I will do it. I will mute every one. Okay. So we will use the organisms. We will put them on the x-axis. You will plot the number of the night time sample. And don't forget that you have two sets of data. You have one for the daytime and one for the night time. So you will plot two sets of data, meaning you will here put the organisms and here you will put the number of organisms. Here number, I didn't continue because it will take me a very long time. Number of organisms on the Y type. And you have to write it like this. Okay, let me number. Here the number of organisms. If you have units, add them. Please mute your mic. You're distracting me and the others. Number, type of organism. Type of organisms. Don't write type of organisms. Write the organisms themselves. So this is wood lice. These are the centipedes. Centipedes. Okay. Then start drawing your pairs. You will do what? You will do one for the daytime and you will do the other one for the night time. For an example, here in this, okay, we have 15 for the wood lice in the daytime. And this is a prepared one that I prepared from so you can see here how you should plot your bar chart. You see here, the blue is for the day, the blue is for the day and the red is for the what? The night. You will draw. So the first one is 7 for the wood lice. During the day, during the night, it's 15. So you see how we make the scale 0, 2, 4, 6, 8, 10, 12. And you will leave a space between each organism and you will do the spiders during the daytime. It's what? During the daytime, it is 1. During the nighttime, it's 2. For the centipedes, there are 2 centipedes during the day and 1 during the night. For the spiders, there are 3 spiders during the daytime and 2 during the what? The night time. These are the labels, name of organisms, and you should put the names here. Here you put number of organisms, and you will add the unit that been mentioned in the question. Here, the number of organisms, to compare the number of organisms collected. They use half of the graph, and here the key is there, the labeling is there, and everything is okay. You will get full mark for this. And you see, no line is done by hand, all of them by ruler. So this is a type of question that you will face in most of the papers. There is another type of questions. I think it's too much for you, but for the benefit of our students, I will continue for more 5 minutes. Okay. This is talking about what? Talking about cloning. How the cloning is done. You have to keep this picture in your brain. Please keep it for as a reference. You need to keep it, to use it. Any question ask you how you clone a mammal, you will use this one. Use it as a reference, because cloning is talking about what? Getting a somatic cell. You will take the nucleus of this somatic cell. It's not a reproductive cell. It's not a sexual cell. It's a somatic cell. It's taking from the body. We will take it from the body of a donor. Donor nucleus. We will take the nucleus. We will try to fuse it with a what? An egg. This egg, it's lacking the nucleus. No nucleus in this egg. With an electrical spark, we will allow the nucleus, the donor nucleus, to be fused with the what? The mother or the other animal that we will take the egg from it. Adult female sheep. So a donor cell is taken from a sheep. You will take only the nucleus, and you will take an egg from an adult female sheep. The nucleus of the egg cell is removed. Then an electrical spark will be used to fuse the nucleus from the donor with the egg, with removed nucleus, and fuse them. The fused cell will start what? Dividing mitotically. Any division that needs to repair or grow, it will use mitotic division. When the embryo become a ball of cells, the embryo will be implanted in a female mother, which called the foster mother. Then it will be a cloned lamb will be produced. So in your opinion, this cloned lamb will be identical or not? From the word clone, it should be identical. And it should be identical to the donor, because the most important part is the nucleus. And this is what happened in sheep, in Dolly the sheep. And this is the what they call it, the usual cloning happening in these organisms. And I wish that I didn't use a lot of charts in my presentation, because I want you to concentrate on the questions and concentrate on the strategies that you have to follow within the past papers to get higher marks. Okay, I'm ready for your questions if you need to ask questions. Okay, Rinad, do you want to ask? Here is one of the questions. It's challenging. I got a lot of questions on my email and on my WhatsApp account via the biology group, and this is one of the questions talking about the saturation of hemoglobin, and most of the questions are asking about, yeah, who wants to, is there any question? Rinat, you want to ask a question? Reading the graphs, you should know your basics. You should know that here. Is there anyone who wants to ask a question? You should know where is the hemoglobin is saturated or where it's unsaturated. Here the area that been saturated, and here that when they start unloading the oxygen, and this is the fetal hemoglobin, and this is the what? This is during a contracted muscle. There is one question, dissociation curve you would for adult hemoglobin, the concentration of carbon dioxide is increased, and this is how it will looks, because with carbon dioxide the rate saturation will be less here, and most of the graphs are talking about enzyme. And here a question for you: eating garlic is thought to reduce the chance. This is one of the investigation questions. Eating garlic is thought to reduce the chance of being bitten by insects, such as mosquito. Design an investigation to find out if people who have eaten garlic are less likely to attract mosquitoes than people who have not eaten garlic. Your answer should include experimental details and be written in full sentences. So I want you to investigate what this case, and you can investigate. Sorry. I want you to apply all the rules that we talked about, and please send it either to my email or to my WhatsApp if you are in the WhatsApp group, because we have an biology O-Level, IGCSE, and AS A-Level group. And this is a demo class for the crash course that I will give during the upcoming months from here till your May and June, and we will continue, of course, and we are giving also normal classes. In the crash courses, we will solve a lot of past papers talking about the different subjects and the different challenges that you are facing. The other thing that the normal classes, it's a normal class. I'm explaining all your subjects. And Dr. Aisha, she will provide you with the links for our platform so you can,
Excuse me?
yeah. Okay, Miss, talk.
Can you send this presentation either to our email or WhatsApp so that we can benefit from the other slides which we didn't do it today?
Yeah, this is not a slide that didn't do it, but it's here.
No,
I have
like you
yeah,
did, you delayed, so because of the time, that's what I said. Like you
no
can send
problem.
it so we
I
can.
will send it. I will send it to you. Are you in the biology group?
Yes.
Okay, so you can send me privately your email, so I will send it. And mostly I will share it with the members of the groups. And here I have one of the past papers. I'm sorry that I couldn't solve, but I want to solve a lot of past papers. But from my experience, I noticed I prefer to summarize the questions. And I think I summarized most of the challenging questions. And the graphs one, there is one with the enzymes inhibition, and you have to know that the graphs with we have two types of inhibitors, the competitive and the non-competitive. So if you have the normal, what we call it, the normal chart, maybe I can use the space. Okay. Okay, I can draw. Yeah, I can draw. You will have the normal. Oh, sorry. My line is not straight. Okay, clear all drawings. You will have the chart like this. So you will have the enzyme going like this. This is the concentration of substrate. I'm sorry for the writing, but I want to, and the rate of enzyme activity. For the one with the inhibitors, you will have the line here. With the non-competitive inhibitor, you will have, you will have the line here. And it will join it again above. And the one with the competitive, it will be above, it will be here. Undo, it would be here. This is the competitive, because for the competitive, if the substrate concentration increased, so at the beginning the rate will slow, then it will go or increase again. But for the non-competitive, it's not affected by the substrate concentration, so it will be here. And here the plateau, because it's the optimum concentration. So after this point, the rate of the enzyme activity is not affected anymore. Thank you. Thank you very much for all of you for being with me. And I wish that I did something to change or help you. I'm here to help you. So any question, provide me with the question. Inshallah, I will be more than ready to help you in solving. Thank you very much. And you can make benefit also of our coming courses and making benefit also of our biology group. Wish you best luck. Thank you all. Thank you very much.
Thank you, doctor. Thank you.
Thank you. Yeah. And Dr. Aisha, she will provide you with the links for our courses, upcoming courses. Wish you the best. Your children will be in our hands. We will help them. We will guide them to do or to get A in their tests. Wish you the best. Thank you very much. You may leave the meeting. Thank you very much. If there is any question, ask it before you are leaving. Okay. Okay.